I am Snehlata Singh, co-founder and chief scientific officer at Biomarker-IQ, where we are developing VarSee Lung, a clinical inference engine to detect lung cancer at its earliest stages. So what you see on your screen is a very unfortunate problem. So currently, more than 90% of the lung cancer cases get detected at stage 3 and 4. The above table is the data from AIIMS Delhi that shows that almost 96% cases are coming in late stages when the survival of chances is less. So how do we increase the chances of survival of these patients? It is by detecting the cancers early. And what we need is a solution that is non-invasive, radiation-free, and can be available across the length and breadth of the country. What we are developing is VarSee Lung, which is a screening tool that is an ELISA-based blood test that measures different biomarkers in the patient blood and puts it into a computational algorithm which then generates a report and clinical insight based on which confirmatory diagnostic tests can be prescribed. So what is our value proposition is that we predict a sensitivity and specificity of more than 90% and an AUC of greater than 0.8. And since our test principle is based on ELISA, it can be adapted by any diagnostic lab which has a basic ELISA machine, and therefore no upgrade of infrastructure is required. What we offer is an ELISA kit with all the antibodies and solutions that you need to measure different biomarker levels, a user interface where you have to input these biomarker levels into the system, and a computational algorithm will run on the cloud and generate a reference report as you can see on your screen. So how did we select our biomarkers? So this biomarker selection was an extensive process involving machine learning as well as scientific expertise. Over the last 1.5 years, we carefully went through more than 3 lakh articles and had our scientific matrices made on which we selected these biomarkers. And finally, we reached 12 biomarkers which are all protein based, and these are predicted to give an early stage sensitivity of more than 92% and specificity of more than 96%. We ruled out cfDNA and CTCs based on their low sensitivity in the early stages as well as technical feasibility in adoption. So now when we came to our 12 biomarker panel that we found out through our extensive data analysis, what we went ahead is with an inpatient validation study where we are currently doing a study in 100 subjects, 50 lung cancer patients and 50 healthy volunteers. And these are our inclusion and exclusion criteria where we have classified lung cancer patients as well as healthy. And our reference standard is tissue biopsy and index test is VarSee score. So I would like to show some of the data that we have obtained till now. We have done these in 40 subjects, 20 lung cancer patients and 20 healthy controls. And what you can see on your screen are 3 different machine learning models that we have run. And with each different iteration of the model, we do see increased performance of the model and ROC curve. So on the extreme left, what you can see is different biomarkers and how much correlation they show with the lung cancer. From this study is still under progress. As you can see, this is just 40 patients and we are currently doing 100, after which we would be able to plot our confusion matrix and get our sensitivity and specificity data. But these subjects we already have, so we plan to finish this study by January 2025. And the next thing that we are planning to do is a multicentric clinical trial to validate our pilot data. And for this, we want to collaborate with Tata Memorial and for recruitment of these patients and conducting this study. So the societal impact of our product is multifold, as timely detection of lung cancer would lead to improved treatment options, reduced cost of healthcare, and increased survival rates. In the last almost 2 years of our incorporation, we have received significant traction, multiple government grants and fellowship. And this is our team being led by Manasi Khasnes, who is a PhD from ISER Pune. Myself, I'm a PhD from NCCS and postdoc from University of Pennsylvania. Atharv is leading our technology team, and he's an expert in machine learning and AI models. We are being advised by Dr. Manoj Kumar Bhatt, distinguished professor and former director NCCS, and Dr. Sasmita Panda, who is an oncologist at Nanavati Hospital. Thank you, and I would love to take your questions.
So while doing the validation, did you take smoking history
Yes.
stratification, and will it be more useful for the smokers
Yes. So
to predict their risk?
right. So currently we have taken age, smoking history, gender, as well as occupational hazard, all these factors into play. So from the 40 subject study that we see right now, we don't see much of association, like significant data is not obtained, but yeah, we are looking into that, and we are looking forward to the complete study, and then maybe we would be able to tell if it is correlated.
Whether the biomarker selection is based on the smoking history?
No, so biomarker selection, when we did take into account age, and so these biomarker levels are not dependent on smoking history, is what we
So
see.
it will be useful for the entire.
Correct. So we have taken into account race, age, gender, smoking history, occupational hazard, alcohol consumption, previous history, so these things. So these are not dependent from our in silico data, we can say.
Thanks for nice presentation. So just wondering about this things about this algorithm, methodological algorithm you are pursuing on
Sorry,
that.
I can.
So methodological algorithm you pursued to have these biomarkers to be selected, as you mentioned that AUC is good enough. So what's that outcome you have chosen to look on that over there?
Okay,
Or
so the
maybe more on the methodological aspect you are looking. Because machine learning gives you so many algorithms, and if you put something, definitely you are going to get something.
Correct. So we are looking at a balance between precision and recall values, and for that we are using basically 6 machine learning algorithms are what we are utilizing currently. So based on that, we'll be like precision and recall values is what we are selecting the biomarkers on.
And in terms of the validations, still we are looking for 1,000 new patients to be validated, not with these 40 patients,
No,
we
so-
don't have any validations cohort.
Correct. So with these 100, we would be able to get our, sensitivity and specificity values that we in lab. But to confirm that and to see that whether if it stands true in a larger data set, we need more data. So I think previous speaker also mentioned that, with machine learning algorithms and AI, you need even more data set. But yes, 1,000 would be something where we will plateau with our sensitivity, and we could be confident that this is what holds true.
Thanks.
It was a similar question. I mean, your sample size is 40 as of now. I was just wondering, with the 40 samples, how did you manage to train your ML? How did you manage to train your machine learning?
Yeah,
model?
so currently what we are doing is we are these 40, we are amplifying to 1,000 data set and then predicting, but we are not able to plot confusion matrix because of that. So
These 40 cases are all cases of early lung cancers.
no, sir. So 40 is basically 20 healthy, 20 lung cancer, and in this we have, I think,
What stage of lung
three
cancer?
stage 1 we have. Rest of them are stage 2 to stage 4.
So stage 2 to stage 4 will not be early lung cancer in
Correct.
any case.
That's true, sir.
Your objective will be to screen.
Yeah,
The screen will be in very early lung cancers.
true.
So whatever model you develop for stage 2 to 4 will not apply to very early lung cancers.
Yes.
And have you identified these biomarkers through immunopeptidomics or what? What has been your tool to reach these 12 biomarkers as your final targets?
So, I'll answer your question in 2 parts. The first part was, I forgot the first part. Okay, I'll go with the second one.
You have a mix of
What's the
one.
So the first part that
My
it would not be.
suggestion is that it should be all one.
So, sir, as I shown in the first slide itself, the problem is that early lung cancer patients are less. So what can be done in this scenario is if you take the later patients and do a logistic regression, and then you have stage 1 few patient, and then you overlap both the data. This is mathematically how we can make sense with even less stage 1 cancer patients and more of stage later. So this is first part. The second part, how we identified these biomarkers, right? For the identification of these biomarkers, what we have done is that we have gone through the scientific databases, and through those 3.5 lakh articles that are published in the lung cancer and diagnostic domains, we have utilized natural language processing, and from there and scientific expertise, we have curated our scores and divided biomarkers into different personas, and then selected with a human in the loop system. And then that we are validating in the lab, last 12.
Is EGFR mutation one of the biomarkers?
No, sir. So these are all serum markers, and yes, so cell-free DNA are comparatively less in earlier stages. So, sorry? Yes. We are taking into account 39, all the 39 subtypes, like even under adenocarcinoma, squamous cell, all the ones. But our clinical data, if I talk about 20, we have from adenocarcinoma and squamous is majorly biased.
So once you're saying about this 17 biomarker, once you're saying about this 17 biomarker, are you making the consolidated scoring system by 17 biomarkers,
Correct.
or making
12.
any thresholding on that? Or
Yes, so these 12 biomarkers in different concentrations. So even for example, some of the biomarkers are present in TB cases as well, in pneumonia cases as well. So these are the differential concentrations and the algorithm, our proprietary algorithm which is running in the back end, that is differentiating between the cancer and the healthy. And by healthy, I mean not cancer. It could be any other lung.
So when you say it is specific, have you compared with some other cancers also, whether it is picking up falsely in that case or not?
So
That is also a good control for you.
that's right, sir. But in our pilot study, we are currently just focusing on lung cancer. And once we solve this problem, then we would be, yeah. Correct, that is. So,
Thank you, Dr. Snehata.
thank you so much.
So we move on to the